week's lecture, but I will do it hopefully today. But it's not related to today's lecture, so it's maybe not so urgent. Actually, the plans initially for this Matter to Life Max Planck uh, school and all the lectures in there were actually that we would be more innovative in terms of lectures anyway, which means more things like inverted classroom, more discussion, more external speakers, not only me giving a traditional lecture here, but in the end, it's difficult to actually really be extremely innovative here. So, at least for me, maybe because I'm too old for it. But in principle, therefore the lecture by Peter Schulz was really in the spirit of how it's supposed to be. So I should also, for example, for cell-free, I could have probably invited other experts. And nobody usually has time. And then the problem is that if you invite different people, then they have time at random times, and then you cannot have an order in the lecture. So that's what I found difficult, because then now we had the Ox DNA lecture in the middle of something completely different in terms of the curriculum. Okay, whatever. So now I filled my last 1.5 minutes and I wanted to switch to a different topic today, which is cell-free synthetic biology. So the initial, well, I think that we are kind of returning to the beginning of the lecture in that sense that initially I was only talking about bio-nanotechnology, which is kind of the easiest part of synthetic biology because you just build molecular structures. So that's kind of easy in comparison. Then most of synthetic biology in the first maybe decade or really focused on working with bacteria, because genetic tools are very well established and they are still relatively simple organisms. But then because of and that was also a question in one of the recent lectures, why many people now work on mammalian cells, or why we would talk a lot about that in some of the lectures. The point is that, of course, that there is a in a sense the field tries to move towards applications, and in the end there are not so many applications out there in principle. So you could either think of reprogramming cells for bioproduction, which means in the context of bacteria, that's the whole metabolic engineering business, where you try to make kind of interesting chemical compounds with bacteria and try to optimize them and these things. But then if you go towards more biomedical applications, then you sort of have to deal with mammalian cells, and therefore many of these other genetic tools and some of the things in terms of, for example, circuits and cellular recording and other things we talked about were more developed for eukaryotic cells, which on the other hand are much more complicated. So they are really kind of complicated and in comparison. So it's really also more difficult, at least as far as I can see it so far, to follow the original vision and program of having a programmable, rational kind of engineering discipline for such systems, because still too many things are probably unknown and too many interactions are going on that cannot be currently de novo sort of designed in a sense. So there's always a lot of screening and still a lot of trial and error approaches, which is not what synthetic biology wanted to be in the beginning. But on the other hand, I have to say that probably things are getting better. It's not that it's hopeless, but it's just much more challenging than probably anticipated in some context. So another approach, and this is what I'm, yes.
That's a little bit off, but now that the protein engineering gets easier and easier with AI and stuff, I mean, how does it challenge the perspective for DNA nanotechnology? Because I mean, in nature, everything is closed and right. So is that maybe bad for DNA nanotechnology, or it doesn't explode, or does not matter?
That's really off topic. So the question is whether DNA nanotechnology is doomed because protein design emerges. Yes and no. I think that I guess that, of course, people would love to do the same things with proteins than what can be done with DNA nanotechnology today. And I think that this will happen soon. But currently, still, the whole workflow for protein design is much more difficult and takes more time than for DNA nanotechnology. So also, it's much easier to do. I think that, for example, if you take a DNA origami structure, everybody in this audience and even school children can learn how to do DNA origami. You just have to learn how to use this cut nano thing. You have to learn that A binds to T and G binds to C, and that's it. Of course, you need to have some characterization tools. So the entry level is relatively low, whereas the protein design business currently requires much more complicated computational resources. And in order to make a protein, you cannot just order the DNA from the supplier and make the structure, but you first have to make a gene that is transcribed and translated, and the protein has to be folded, and then protein characterization is complicated. So just the overall workflow is just much more complicated. So therefore, but on the other hand, of course, the problem is that maybe DNA nanostructures are useless. So in the end, you're doing a lot of kind of crazy stuff, but there's no real application for it. So I think that this will be decided in the future, how far you can get with DNA nanotechnology, and whether, for example, any insight that you can get from DNA nanotech can be used for other things. So one argument often is made for prototyping, that one says, I'm first going to prototype something before I'm going to do the real thing. But then, of course, the question is whether DNA has anything to do with protein in a sense. So I guess that one thing that can be done is explore fundamental principles of self-assembly. So maybe some of the things can be used. So that's what I think. Then in other cases where actually DNA can be used, maybe because also DNA is relatively stable, so it can maybe sustain other environmental conditions than maybe more fragile proteins. Maybe there are applications where the proteins are actually not working so well, and that could actually be non-biological applications. Let's say if people want to make like sensors or use them in a more nanotechnology context. And of course, then there's this intermediate direction that many people work on at the moment. It would be RNA nanotechnology, and I think that many people try to, of course, go from DNA to RNA. And in a cellular context, RNA then interacts with proteins. And what you have to do in the end is just do everything. So I think that the future probably is just trying to design biomolecular systems from all these components, and then hopefully the tools will be kind of advanced enough so that you can sort of choose the right weapon for the task. I think that's a very long answer to you. So it's vaguely related to the lecture, because here the argument is that cells are too complicated, and maybe some things can be done easier in a cell-free context. And there is also a big or a growing community of cell-free synthetic biology. And one of the problems with getting, for example, synthetic gene circuits to work in cells is that whatever you do, whatever you create as a synthetic circuit somehow interacts with the living cell. And in a sense, you have a different goal than the cell has. So the cell wants to grow, and that's that quote here by Jacob, that the cell, the only goal of a cell is to grow and divide. And now you introduce some synthetic components, and these obviously disturb, or they usually disturb the cell. So it disturbs the cell in its goal to grow and divide, because it has to do something that it is actually not interested in. And then, of course, in cloning, you have to come up with tricks. So you put antibiotics resistances on the plasmids, otherwise the bacteria would just get rid of them because they don't want to express the foreign gene in the first place. So one thing would be to just get rid of the cell altogether. So we are just interested in the biochemical machinery, and we want to kind of just perform certain biochemical tasks in a cell. And one possibility would be to just not work with cells and reconstruct something that is similar to a cell but doesn't grow, etc. The other thing, of course, is that cells are compartments, and and sometimes it's difficult to get components into the cell across the membrane. And the other thing is that in certain contexts, you are actually also interested in just mixing components from different origin. Maybe you want to have a gene from one bacterium and mix it with something from a mammalian cell, and maybe also some completely synthetic compound. You might have a chemical biologist might have produced something for you, and you want to mix it with your reaction. Then it's difficult to deliver all these different components into a cell, and maybe it's easier to just have a reaction chamber that contains all these components, and which would be kind of a hybrid system in the end. So therefore, there are kind of reasons that why cell-free approaches are interesting. On the other hand, and that's the drawback, and that's actually the same drawback as the advantages, that cell-free systems don't grow and divide. So therefore, one of the challenges for this field is that there is no metabolism at the moment that would allow you to run these systems forever. And that there is no equivalent to a real bacterial culture, for example, at the moment. So it's difficult to think about these things. But this is something that many people work on at the moment. So here is just a brief history, and cell-free systems actually have been already produced a long time ago. Here in the 1950s, people actually took just extracts from cells or tissues and looked, for example, here they just saw that when they add radioactively labeled amino acids to such a kind of extract, that these amino acids would be integrated into the proteins somehow, that if they were analyzed after the reaction. That showed that there is some kind of metabolism and protein production going on in these cells, in these systems, even after they have been after the cells have been broken up. And then that's kind of a famous example that is always cited. Nirenberg and Mattei actually elucidated the genetic code based on cell-free reactions. And I have a slide on the next slide, so I'm going to talk more about that. And then there have been actually many developments where people try to make these extracts from cells more efficient and figured out what would be the optimal recipe for it. Here is an important step. This Shimizu paper in 2001 is the so-called pure system. that is a system that contains only purified components. So, and I'm coming back to that later. So in contrast to a cell extract where you just break open the cell membrane essentially or wall in the bacteria, you have in these extracts, you have all the components in there, and it's not a very well-defined system. And the pure system, they just extracted all the necessary components for transcription and translation, purified them, and then put them together again. So there's nothing else in this system, or almost nothing else. And the popular recipe for making cell extract is the MyTXTL recipe that was published, for example, in this paper here. And I'm going to talk about these two systems because they are often used by many labs. So here's this is a picture of Mattei and Nierenberg, and they actually tried to figure out what nucleic acid codons or which nucleic acid sequences are responsible for making proteins. And apparently, like here in the early 1960s or end of the 50s, it was not, so mRNA was not known as a molecule. There was only a hypothesis that it existed and it had some role in gene expression. So what they tried to do is they made synthetic RNA molecules and then added them to cell extracts to see what happens. So there was a hypothesis that it has something to do with protein synthesis. And that apparently was a major kind of experimental effort. And Mattei here actually prepared radioactively labeled amino acids. And what is written here is that he essentially used growth media for the bacteria that only contained C14 carbon. And so they incorporated all the C14 carbon in everything that they made these bacteria, including the amino acids. So you can then extract or purify the amino acids, and they all only contain C14. And then they made experiments with mixtures of sort of amino acids that were radioactive or not, and others. And they also synthesized simple polynucleotides. And from that, they could then essentially see which polynucleotide sequence coded for which amino acid. As an example here, so that's counts per minute per milligram protein. So what they had a crude extract from E. coli, so just breaking open the cells, and they added poly-U, so an RNA molecule that is just U, so like a poly-U, and then they added phenylaniline to the solution, and they saw that there was the cell extract produced polyphenylaniline that they could from which they could measure the radioactivity. So the signal went up here, and if they added other amino acids, nothing happened. So that essentially says if I have poly-U, then phenylaniline is incorporated into amino acids, and from that and this is the generated that table here. So poly-U gives phenylaniline, or these are incorporated. That's what the table means. And here proline, lysine. But then they also tried more kind of complicated systems. For example, adding U and C at the same time in the polynucleotides, and that actually generates sequences like you can have UC or UUC, UCU, and so on, all kinds of permutations. And so they, and if you look at a table for the genetic code that of course was elucidated later, you will figure out that with these combinations, you can code for leucine and serine. UA gives you more possibilities. And from these experiments, they could sort of construct how the genetic code looks like. Here's also an interesting experiment. So they looked at phenylalanine incorporated when they actually, here they have a poly-U RNA molecule, and they added a poly-C, and that leads to still kind of production of this polyphenylalanine. And when they add poly-A, this drops. And the point is that, of course, we know that poly-A base pairs with poly-U. So in the end, you will have a double helical RNA molecule that is not translated into proteins. And from that, they figured out that the mRNA must be a single-stranded molecule. So otherwise, the double strand would not work. So that was kind of a detective story that was really only possible with having the cell extract as the kind of containing the machinery for making proteins. And here is a recent review by Michael Jewett, who is one of the best known people, I guess, in this field now. He just recently got a job in Stanford, so he made it made a big career with cell-free synthetic biology, I guess. And here is a timeline that they showed. And here again is this initial start with the Nirenberg experiment shown here. And then here's actually another, here's the pure express, here's another milestone where Noah Rowe and Liebschaber first incorporated actually a cell-free expression system in a lipid vesicle, and then they called it an artificial cell or something like this. So that was kind of the beginning of this type of research. And then production of bacteriophages is a milestone. So you can actually add the genome of a bacteriophage to a cell extract, and the cell extract will make bacteriophages. That's actually quite impressive because you need to consider that not only the proteins are produced by the phage, but the whole thing happens in the cell extract. So the proteins are produced, they package the genome of the phage, and the phage is self-assembled, and everything outside of a bacterium. So it's in a sense, it's a rather complex self-assembly process that actually works also outside of the cell. And while the other applications maybe are not so important, and I'm coming back to that later anyway. So there are now many kind of different ideas what one could do with cell-free systems. So I already mentioned prototyping earlier, so this is also often used for prototyping of genetic circuits. Then there's a lot of interest in cell-free production of valuable compounds, metabolic engineering, or using these systems as sensitive biosensors, maybe for also diagnostics. And mixed chemistries is one of the kind of big advantages, as I said already in the beginning, that you can really compose your system from very different sources, which is easier than in a cellular context. And of course, synthetic cells, they use these types of systems a lot, but that would be the topic of the next lecture. So therefore, I'm not going to talk about synthetic cells now. And then here, basic biology, biophysics. I think in the end, it's also, of course, interesting to look at these systems as simply reduced biochemical systems where you have a better knowledge about the contents of the system and might be able to really kind of understand a little more how exactly the processes in the system occur. So here, also from this review paper that is cited down here, there are many kind of examples how that could be used. So prototyping. One example is that this is kind of our typical gene expression setup that we talked a lot about in this lecture. For example, here's a promoter, here's a ribosome binding site, coding sequence terminator, and here you see there's N. That means that could be, for example, a randomized sequence region in the promoter region. So you might look for a transcription factor or maybe a different an evolved polymerase or something like this that would bind to this specific sequence. And then you could actually make a library of these sequences. So you will have many different possibilities, and then check which of them sort of produces more protein or less protein. So this could be done, of course, also in cells. You could put that on a plasmid and clone it and then look at the fluorescence in cells. Or you could say, well, I don't want to go through the pain of cloning and so on. You can just put a linear, and this is why it's called linear expression template. So you just use the linear template and then put it in a cell extract and check how much fluorescence is generated. There's actually a problem with linear templates, and that actually in a crude extract, you have enzymes that actually destroy DNA molecules because there are exonucleases that chew up the DNA. So you need to somehow protect them. Or if you use the pure system, they don't contain these nucleases. So that's maybe more a technical thing. But then here's one example where this is actually then done in droplet microfluidics. So you can actually put together the reaction mix and then check the better performing or look at the fluorescence of the droplets and sort out the ones that work better. And then you need to analyze the sorted population and maybe sequence them and figure out what sequences actually generated a better result. In principle, the same thing as here, but in a more complicated thing can be done here, where you would actually prototype gene circuits. That is more complicated because if you look, for example, at dynamical circuits like this here, you cannot do it, for example, in a droplet because you would need to observe it for a longer time. So it's just technically more complicated. But what you can do in principle, and this is, I think we haven't really talked a lot about that, except in this one lecture on the design of gene circuits with this computer program. If you remember, there was that program Cello that allowed you to match output and input relations. So you could also try to match, for example, the output and the input of two genetic parts by using libraries where you try to optimize the coupling between two genetic elements. So the advantage of cell-free systems is summarized here. So the idea is that, as I already said, that this would be much faster if you do a cell-free approach. Here would be your expression template. And here the arrow says you just do the experiment after you got that DNA. Whereas in the other case, you need to go through the cloning step. And depending on how complicated that is, you might actually save a lot of time going just through this, just using cell-free experiments. The problem, on the other hand, of course, is that it's not always the case, unfortunately, that an experiment that works in cell-free also works in the cell. So that's, or maybe they work very differently. And this is still one of the challenges and also mysteries, how to exactly relate the two. And that has to do with the fact that actually, of course, the growth of the cells has something to do with the influences the dynamics. And another point that plays a role is, for example, spatial organization. The cytosol in a bacterium is much more concentrated than a typical cell extract, and therefore concentration dependent effects and also crowding and other things play a role in gene expression, and that could lead to very different behaviors in the two systems, and that's still a challenge. But you see that this is somehow, in a sense, it's related to the goal to create synthetic cells, in that sense that, of course, you would like to create a system that behaves exactly like a cell. So you could then transfer the results. And for this, there is still maybe some kind of unknown points that need to be clarified. On a more practical level, of course, you can use these systems for, just because it's a convenient system to make proteins. And here is an example where the idea is that you could actually use also orthogonal tRNAs, which allows you to actually incorporate non-canonical amino acids. I'm not sure whether I have time to talk about this topic in the lecture, but there are efforts to expand the genetic code, so you can also introduce foreign or like non-natural amino acids into cells, and that could also be done in a cell-free context. Here, enzymatic pathway prototyping. In enzymatic pathways, the problem often is that you actually have a series of catalytic steps, and the overall metabolic flux depends on the ratio between these, because if the enzymes have a different, let's say, speed, then the problem sometimes arises that one enzyme is just too fast or too slow, and then there is an accumulation of intermediate products, and you want to exactly match, in a sense, everything, so that everything that is produced by one enzyme is used up at the same rate by the next enzyme. And to screen for the optimum is something that is important for these tasks. That's one possibility. Protein-protein interactions, but in principle, you can of course do everything that you can imagine that requires kind of simple production of proteins and interactions. Here is a different example in terms of sensing. So one example that we already had in the lecture a little earlier are these so-called toehold switches. These are RNA molecules that can be switched from a translation inactive to a translational active state, or vice versa. And this has been used for biosensing. In principle, the RNA that comes in here, the trigger RNA, sort of opens that structure here and then activates a gene expression. And if this gene expression is something like a fluorescent protein, for example, you could use that as a readout for the detection of a specific RNA. And so that would be the sensing kind of scheme. And here the transcription and translation processes are simply amplification processes. So they just make many sort of molecules out of one sort of detection event. And in this context, what has been done is, for example, make freeze-dried cell extract. So you can just take the cell extract from bacteria, then put them on paper, freeze-dry them, and then you can rehydrate them by putting some water or buffer on top, and then that would be an active protein-producing extract again. So you can, in principle, use that in a sense as for test stripes if you want. Yeah, here, well, of course, you can also use them for small molecule sensors, essentially because certain transcription factors are also respond to certain chemicals that you are interested in, and that can be used. For example, there are, let's say, mercury sensors, so there are mercury dependent transcription factors, so you could in principle use that, or maybe whatever comes to your mind. If you have the components. So in this context, you could always say, well, I can also detect these molecules in a different way. But if you happen to have such a system where a transcription factor responds to a molecule, then you get this genetic process here as an amplification mechanism in principle. Yeah, biomanufacturing, in a sense, I already talked about that before. Here would be the orthogonal tRNA non-canonical amino acids. You can make different proteins with that. And here, glycosylation, that is something that is of interest because many proteins that you need in a biomedical context for applications in mammalian systems, for example, are glycosylated proteins. And these proteins, but it's difficult to make that in E. coli. On the other hand, it's easy to make proteins with the cell extract, and what you can do in principle is modify the extract in such a way that it produces the proteins, but also contains the glycosylation machinery, and that then allows you to make glycosylated proteins in a cell extract, which then has biomedical relevance. So that's just a survey of things that. So this sounds like cell-free synthetic biology is great, of course, but on the other hand, there are drawbacks like these systems don't run forever, and you cannot do everything. It's maybe in particular because they don't run for a very long time, you cannot make very complicated things like a circuit that takes a very long time for completion is difficult to make, or dynamical systems are difficult to implement. But for protein production and such tasks, it's certainly a very good option. Yeah.
For example, in chemical manufacturing, you also use like a flow jet reactor.
Mm-hmm.
So I mean, you can also use and then you can run it forever.
Yeah. Things like this have been done, and I come back to that later. But so I'm not an expert in this kind of scaling up of such processes. The only point is that for making cell extract, you first have to make bacteria. So you can, of course, grow the bacteria in a bioreactor, and then you have to mesh them and extract everything. And so the amount is not extremely large. So it's not on a, as far as I know, it's not on a liter or hundreds of liters scale. So that's not so easy to do on a really large scale. But I would not rule out that it can be done at one point. And I guess therefore some people would like to focus probably more on high, let's say, sometimes the biosynthesis people in a sense distinguish between high value chemicals where you need small amounts that and they are extremely expensive to make. So then it's more useful to have like a small scale synthesis, whereas things where you need like, I don't know, kilograms of material as the product, that's kind of more challenging. And then it's probably easier to really do it with E. coli and make a big fermenter or so. So it depends a little on the application. So here I now want to be a slightly more technical. So how are the extracts actually really made? There are these crude cell lysates where you just destroy the cell membrane or wall and then take what you get. Then there is cell extracts. So that's only lysate. That's the extract only contains some of the components that you're interested in. And you could also purify these components. And depending on through how much effort you go, you get more or less defined systems and more or less volume and so on. And yeah, here, when you do these things, you need to think about what you actually want to achieve. Do you only want to produce proteins, or do you want to see dynamics? Do you want to mimic cellular biology, or you don't care and it's just a production sort of only the production machinery is of interest and so on. And therefore, some of the systems are better suited for certain things than others. For example, a pure system does not mimic exactly what is going on in E. coli because all the other components are kind of missing. And then it might be actually better to use a cell extract, which contains kind of more components. But on the other hand, the cell extract also, as I said, contains degradation proteins, which would then reduce, for example, your yield or influence the dynamics of the expression profile. So it depends on what you want in the end. So the production of the cell lysate, that is with various uses various brute force approaches, like you just mash them with these so-called homogenizers. You press them like under high pressure through an opening. That's the famous French press. And there's a machine that kind of just shakes your cells with little beads. You can also use ultrasound. There are many different methods, and all of them somehow influence the quality of your final result, because some of these things actually disrupt membranes, others don't disrupt membranes so much. And some lead to denaturation of the proteins because there's often heating effects. For example, with ultrasound, you need to put everything on ice and like all kinds of things. And no matter what you do in the end, you have somehow kind of influenced the contents of the cell so that the resulting extracts are more or less active. And of course, people have tried to optimize these protocols, and in some cases, actually analyze what you get afterwards. So also Vincent Noirot, but also in our group, we did some work with people from mass spectroscopy, where you check afterwards which proteins are actually still in there. And it's not very easy to understand, I have to say, what's going on. So a very popular protocol is shown here. That's by Noirot and co-workers. And here they first destroyed the cells. Then there is a centrifugation step, and that is actually already present in a protocol from the 70s. That's often known as the Subai protocol or extract. And that is called S30 extract, which has to do something with the centrifugation speed. So essentially, during the centrifugation, some of the components are obviously sedimenting and others are still in the supernatant. And in the supernatant, there are apparently the ribosomes and all the proteins that you need for transcription, translation, but no DNA or small molecule components. And then, so what this extract is supposed to give you is essentially the macromolecular components that you need. And but then you're missing all kind of the small molecules that you need for making RNA and proteins. So you need to add NTPs, tRNA, the amino acids, and other molecules that are kind of important during all these reactions, like NAD. And then you need to add your DNA, because the DNA is not left in the cell extract, and then whatever is on the DNA ideally is expressed in the extract. So here is one of the few papers that investigated the kinetics of the reactions in the system in more detail, and that is a very unusual physical review letters paper, because usually you don't find papers like this in PRL, but they somehow sent it there. And I think that the most important statements for us in this lecture is that essentially what is written here, so that TXTL, transcription, translation, machinery, concentration, and rates are 1 to 2 orders of magnitude lower than in vivo values. So that is an important point. So what the cell extract are simply less active than what you get in an E. coli. And that, of course, plays a role when you are thinking of dynamical circuits. So if everything is just 1 or 2 orders slower, then some things will not work anymore, or they will work differently. And that's consistent with volume dilution from E. coli. So E. coli, they state, has a mass density of 300 milligram per milliliter of the the cell extract only 10 milligram per milliliter. So that would be a dilution of the macromolecular content by a factor of 30. And here are some rates. So they say that you get transcription speeds of incorporation of 1 nucleotide per second, and translation rate of several amino acids per second. And with these numbers, one can, in principle, make rough estimates. So here they also claim that you have ribosomes in a concentration of 30 nanomolar or more. And some of these numbers differ very much from previous references that they cite in the paper, but they claim that they made really very thoughtful experiments. So the only thing we need to remember, it's less active than in the cell. So so that would relate to kind of this type of extract, and the pure system is this kind of alternative that you can use with well-defined components, and the pure system is shown here. It only contains what you need for transcription translation. So for transcription, you need an RNA polymerase. And in the case of the pure system, they use T7 RNA polymerase. That's an important difference to the E. coli extract, because in E. coli extract, you have E. coli polymerase, which is much slower than T7, also has a different promoter or different promoters, and so on. So that's something to consider. So RNA polymerase, the T7 then makes RNA, and for making proteins out of RNA, you need a lot of components. So you need the ribosome, but you also need the tRNAs. The tRNAs are charged with amino acids. And unfortunately, the amino acid bond to the tRNA is unstable. So you cannot prepare them before. So they are actually produced inside of the extract. And for this, you have the amino acid tRNA synthetases. So they need to be present in the solution. So the naked tRNAs are charged with amino acids in the system in this process. And then you have the ribosome, and that makes the proteins. And for the translation itself, you also need the initiation factor, the elongation factors, the ribosomal release factor and the release factors and the ribosomal recovery factors. So that is even though it's a reduced system, it still is quite a number of components that you need, and then you add essentially the amino acids, the nucleotides, and some small molecule stuff like ATP and creatine phosphate, that's for energy generation, and this hydrofolic acid and a few enzymes here. And I come back to the role of these enzymes later. So the point is that all of these processes actually use ATP and GTP all the time. And therefore, you also have a few processes like the creatine kinase, for example, produces ATP from creatine phosphate, as far as I know. And so there's some process that sort of regenerates the ATP. So here, so I guess that's maybe also interesting when you really work a lot with these kind of cell-free systems. You also learn a lot about biochemistry in that sense that the related question, of course, is what is the minimum number of components that I need to run a biochemical system? So what is the absolutely essential kind of set of components that you need, which also is related to this kind of other question of how to create synthetic living systems maybe at one point. And there are different angles to approach that. And so here, the TXTL extract is composed of three kind of parts. So one contains the cell extract that, and one contains the DNA, and then the buffer, and the buffers. So you need to have the amino acids. So apparently you need a little less leucine than the other amino acids, and the energy solution. And here you see that HEPIS, that's just maybe makes sort of stabilizes the pH in your system. And then you have the NTPs, and you see that you need a little more ATP and GTP than the others, because on the one hand, ATP, GTP, well, all these nucleotides are incorporated in RNA, but you also need ATP for some energy consuming processes, and GTP is also used up during translation because, for example, the elongation factor 2 hydrolyzes one GTP while a tRNA is incorporated. Maybe you remember from the lecture. So you need these in slightly larger concentrations. Then the tRNAs, spermidine is often found in many kind of such compositions. Spermidine can actually enhance interactions between proteins and DNA, or kind of perform or condense DNA and do kind of other things. And then you can add something like an energy source, and this is in this case phosphoglyceric acid. And there is some metabolic process going on in the system that actually generates additional ATP from this. But there are different recipes where people add different things. You can also add phosphoenol pyruvate, creatine phosphate, and all these things. And I'm not really an expert in metabolism, so I can't really say too much about that. And PEG as a crowding agent to at least mimic a little the more crowded intracellular environment of E. coli. So that is that. And here the point is that this crude extract, or the S30 extract probably, is something that contains many components that you don't really know or that are not very well characterized. And that is the difference to the pure system. So the pure system, in contrast, has this list of macromolecules in there. And this is the initiation factors, elongation factors, release factors, and then all the amino acid tRNA synthetases that you need. And here is, in addition, ribosomes. So the ribosomes need to be, for the pure express, need to be purified from cells and added. And then a few enzymes and the T7 enzymes for metabolism and the T7 RNA polymerase for making RNA. And then the small kind of components here, again, ATP and GTP in excess, and creatine phosphate for energy regeneration. So we already had these other things. So that shows you, in a sense, how complicated or how easy it is. So in a sense, if you really want to do transcription translation, you probably need this amount of components and not less. So the question is whether one could come up maybe with an alternative biochemistry that uses less components, but that is not clear, I would say. So one, of course, obviously you need a lot of these here because you need all the amino acids. So if you want to make maybe proteins with less amino acids, with a different set of amino acids, if that's possible. So maybe here protein design could come in. You tell your AI, make my protein only with these 10 amino acids, and then maybe you can reduce that a little. I don't know. So unfortunately, the pure express, even though it's because it's better defined, is relatively expensive. And I actually checked this morning; it's more expensive than on that slide. It's more than $200 now for 250 microliters. And so you have the kind of these options. Either you have this relatively cheap cell extract where you just grow bacteria and mash up everything yourself. There is also a commercial supplier for the TXTL, or you use the sort of better defined pure express, but that's relatively expensive. So the hope is that one can actually produce kind of the recipes that make better defined extracts more cheaply. And here's an interesting paper by Sebastian Merkel. And what they did is they went through a major effort and actually tagged all the components of the cell extract, of the pure system with histidine. So all the synthetases here, the initiation factors, and so on. And then they put like the code for these histidine tagged proteins onto plasmids. And then each plasmid was put into a different bacterial culture. So in the end, you would get 36 different types of bacteria, and each bacterium is responsible only for producing one type of these proteins. And then you could, after the bacteria have grown and produced the proteins, you can extract these components via the histidine tag. So there's some affinity type of extraction. Yeah, that's one possibility. And with that, they could actually reduce the cost to, they say, $0.09 per microliter. So you need to multiply that with 250 and compare with this number, but it's considerably cheaper. Yeah. So that's at least promising that this might be, well, could give you cheap or less expensive extracts if you want to scale things up at one point. one thing that is still not done here is actually they don't produce the ribosomes, and that's one of the big challenges still. And I'm coming back to at the end. So you always need to add the ribosomes from some other purification step. So one other thing is, of course, here you see that, well, you have like these types of concentrations for the components, and you have different concentrations here and different mixtures, and it's not really clear what is the best protocol. And so there are big differences between the protocols. So one maybe obvious idea nowadays is to use machine learning to optimize that. And here's one of the earlier papers on that. And there is, but there are still efforts going on. And what they did here is they chose 11 small molecule components. So these are really small molecules, only amino acid, tRNA, folinic acid, spermidine, and not the macromolecular content. And they had 4 different types of concentrations. So like small, 2 intermediate, and 1 large. And that gives you, with 11 components, 4 million possible compositions. And they tried to sort of find a good composition of these small molecules. And what they did here is they went through one of these kind of learning cycles, including some machine learning here. And the idea is, in principle, that you make kind of different mixtures of these, and they only had 102 compositions in the beginning. And here are depicted kind of neural networks where you have the 11 concentrations as inputs, and the output would be how much GFP does my system generate based on that mixture. So what they then did is they actually pipetted all these different mixtures. And this is indicated here. That's one of these high throughput droplet dispensing systems that allows you to make many different mixtures. Then they looked at the fluorescence, measured that, and then they could feed back kind of the fluorescence values for all these different mixtures into the neural network, and that adjusted kind of all the weights as usual. And from that they tried to optimize the protocol. So they claim that their yield through that process went up over several iterations. And maybe the interesting thing is that here they found in the end, they tried to figure out what is particularly important in this in this mixture. So they have this graph shows you the dependence between the yield and the components concentration, and they make here a strange they use as a measure for that the mutual information score, and essentially that means so how related is kind of the output to a variation, or the variation in the output to a variation in the input. And the interesting thing is that they found in certain cases that it doesn't matter at all what you do. So when you change the concentrations, and one of them is, for example, folinic acid here, doesn't seem to have a big impact. And I think that they even found out that you can even get rid of folinic acid and it doesn't matter. So the reason why people add folinic acid to the reaction is that apparently this is also required to make I guess it's involved in making the F-methionine that is usually used as the first amino acid during translation, and apparently this is not really necessary, but I'm not completely sure about that. The point is that they only use these small molecules here, and in my experience or in our experience, that one of the issues is that it's kind of easy to screen these things because they are aqueous solutions of some kind of small molecule. But the higher, like some of the other components of the extract, they are more viscous solutions, and they are really ugly to handle with pipettes, but also with lab automation, with automated pipetting and whatever. And it simply doesn't work. So the point is that it's too sticky, and but you cannot dilute these things because otherwise your yield goes down when you dilute all kind of the macromolecular content. So that's actually a challenge for screening. So therefore it's kind of easy to do this, but you cannot really screen the macromolecular content so easily. Another question is the metabolism. So as I already said, all the processes use up ATP, but also GTP, and there are investigations into this. A lot has been done actually by Jim Swartz, who was the supervisor of Mike Jewett in the past. And so they claim that when you make a cell extract from E. coli, then somehow you end up with small vesicles inside of your extract, and the vesicle still contains F-ATPase. And maybe you remember that from biochemistry, that F-ATPase is responsible for making ATP, and in E. coli it sits in the membrane. But when you destroy the membrane during kind of the extract preparation, you end up with little lipid membranes like this here. So they claim that in the system still these reactions can go on. And there is, for example, glutamate goes through kind of this type of reaction here. It generates NADH, and NADH is actually used by this electron transport chain to pump hydrogen across the membrane. And that would generate actually a proton gradient across that membrane. So the protons flow through F-ATPase and produce ATP. And that still goes on in the extract even after the cells have died. And so this would kind of help you to run these systems for longer. But there are other tricks that try to extend the lifetimes of the extract. For example, adding maltose is a trick. And maltose is a disaccharide that actually of glucose, and if you split that, you get glucose and glucose phosphate, and so then from that you get glucose 6 phosphate, fructose 1.65 phosphate, and that actually uses up ATP, these steps here. And but here there's kind of glycolysis. You get back 1 ATP, and here in that step you get back 2 ATP. So the overall balance here is that you use 2 ATP, but you get 3 ATP. So you get 1 ATP when you go through that cycle when you add maltose. So apparently and they show that this actually increases the yield. Here you see GFP over time. When you add no maltose, that's the lowest curve. When you add maltose, it's getting higher. So apparently this helps. So there are kind of many things like this which still are waiting for optimization. So now coming back to one of the comments earlier, of course, you can think of trying to run these systems longer by making them in some type of like flow reactor setup. So the batch reaction would be the simplest setup where just everything is used up and then at one point it stops. There are other setups like, for example, these two here, where you have some feeding buffer and that separates your reaction either by a membrane or some kind of other interface. And that allows you to exchange small molecules or also macromolecules with this kind of feeding solution to extend the reaction. That there are actually also specialized systems, and I'm not talking about that. If you want to express, for example, membrane proteins, you also need membranes, because the membrane proteins, usually the membrane bound part is hydrophobic, or so the ribosomes insert the proteins into the membrane during translation, or they fold maybe during translation in the membrane. So you need to sort of also consider that in the reaction. And here would be the continuous flow setup, where you just pump in stuff all the time and you pump out stuff. And if you don't want to lose all your macromolecules, like the ribosomes, but also the protein products, then you have to put a filter in here. So in terms of circuits, there has been some work on cell-free circuits, and because of what I just said and earlier, the problem with circuits is that they sometimes take a very long time. So if your circuit takes 12 hours to complete or to run, then your cell extract is simply already exhausted, sort of before the circuit can actually be really executed. And that is related to the problem that the extract is less active than the expression machinery in E. coli because of this dilution. So everything is slower anyway, and then it doesn't run for a very long time. And therefore, to really mimic like more complicated circuits, people have to use microfluidic setups to pump through the extract and energy solutions and so on for longer. So one setup is shown here that was developed by Sebastian Merkel's group with Henrike Nieder-Holtmeier. By the way, Henrike Nieder-Holtmeier now is a colleague at TU Munich. She was hired last year, but she's in the campus Straubing. But she's really an expert in microfluidics and cell-free gene expression, and also artificial cell type of research. And in this setup, the idea is that you have a microfluidic system that contains such a ring chamber, and the ring chamber can be pinched off from the surroundings via valves, and then you pump whatever you have in this with a peristaltic pump. So these little things here are peristaltic pumps, so you can pump the solution, and in this you can perform cell-free expression. But then after some time, your cell extract has been used up, then you can actually open the valves here. So this valve here and this valve, and you push in new transcription translation mix from here. That's the yellow stuff. And the exhausted stuff goes out here. Then you close the valves again. Now you have your original solution and a fraction of the new extract. Then you can pump that in a circle. Okay, here you can also add DNA. When you have a system that actually allows you to both mix in DNA and transcription translation system. And then you kind of mix everything, and then the reaction goes on. Of course, you always lose something here. So you lose a little of the product here, but you flush in new material. And that is something that you need to live with. But actually, this sort of mimics bacterial growth and division. So bacteria also sort of, they grow in size and then they divide. And this is actually, in a sense, similar. So it would be like your bacterium grows here and then it divides by splitting off stuff, in a sense. And really, by timing the addition of new extract and new DNA, you can actually mimic the dilution of the reactor, so that the dilution of the reactor can mimic bacterial growth. So you can make a dilution rate that exactly corresponds to the growth in a bacterium, for example. So these are relatively complicated microfluidic systems. So you need 2 layers of PDMS, where one layer is used as a control layer that pinches off the fluid layers in the that carry all the fluids. And here, what you see is that it actually works. It's probably difficult to understand what is going on here, but maybe you remember from our lectures where we talked about quantitative modeling of RNA and protein production. There's always this production rate, alpha, and some degradation rate, and you will have a steady state in the simplest case, that is where the concentration of your RNA or protein is given by the production rate divided by the degradation rate. So you have it's this alpha divided by delta. And what you see here is that there's a reaction volume replaced per 15 minutes. And that is something like the dilution rate. So if you dilute quickly, if you, for example, replace 40% of the whole volume every 15 minutes, that's a faster dilution than if you only replace 16%. And here you see that's the RNA and the protein concentration. It goes up, and here is some kind of steady state. And the steady state is higher when the dilution rate, that's our delta, is smaller. So that is kind of consistent. But what is interesting now is that here the reaction actually is monitored for 30 hours. So that can really now run for a very long time. And you can also vary kind of things. So I'm not going, so this is, they just added the DNA in pulses here. So the DNA is added, and then this influences gene expression. So that these are not really circuits, it's only production. But here they actually use the same system to test the repressilator in a cell-free system. And the repressilator we had in the oscillation lecture, and they tried to get that running in such a microfluidic reactor, and that's really challenging because you need to operate the system for a very long time. And here, that only shows the so-called design build test cycle, where you just make your DNA, and then you look whether it works or not, and then you try to go through that cycle several times. And they claim that in their approach using cell-free systems, you can do the things much faster than when you work with E. coli. So here they claim, for example, that you can go using the cell-free approach, you can get your results within a few days, and it might take much longer when you do it with cells. And what they did, actually, they adopted the repressilator topology, but with different transcription factors. So these are, in the repressilator, it was TET-R lambda repressor and LUC repressor, if you remember. And here it's TET-R and these other guys. And they show that also with these, you can actually get oscillations in the cell-free system. And what is remarkable, and that's the only paper, at least that I know, that they also did that with 5 nodes. So remember, we said for making oscillations, you need, in a sense, a negative feedback circuit. And in order to get negative feedback with repression, you need either 3 repressions, but you could also use 5 repressions, like an odd number of repressions. And this actually works, and that's kind of amazing that they got that to run. And they actually, in the paper, also show that they could even then transplant the circuit in E. coli, and it also still worked, which is actually surprising. Yeah, so that's that. Here's a very different approach by Roy Bassif. So I think that not many people use the same approach, but rather than making this pumping system, they came up with the idea to immobilize all the DNA on a chip surface, and that allows them to essentially flush in all the reactants, and the DNA is then transcribed and translated in the reactor, and it is not flushed out by the fluidic system. Of course, the point is that all the proteins that are produced in this process, they are kind of lost by microfluidics. So in the end, in such a system, you have a steady state of the production, or not a steady state, but you have a balance of production of the proteins and the loss in the fluidic system. And what is interesting in this specific paper, they made a fluidic system that looks like this here. Here's a kind of a channel that supplies cell extract all the time. So the components of the cell extract can diffuse through the channel into these chambers that contain the DNA. Then RNA polymerase produces RNA, and the RNA then is actually translated into proteins. So in principle, the RNA can diffuse out again, but also the proteins can diffuse out. So everything can diffuse out, obviously, but it depends on the geometry of that channel, how quick that occurs. So there will be some type of diffusion profile when you look at the chamber here. So there will be a high concentration of proteins and RNA here. And because the concentration in the fluidic system essentially is zero, then when you solve the diffusion equation, it just gives you a straight line from the concentration in here to zero concentration in the channel. And that also shows you that by changing the length here, you can actually change, as in a sense, the lifetime of the DNA in this chamber, because not the DNA, the lifetime of RNA and protein in the chamber. Because when you have a very short channel, then everything that's produced here is just very quickly lost. If it's a long channel, it stays there much longer. And they then put different gene circuits onto that brush. And here you see an oscillation circuit. So in this case, activation and repression. And it also actually works in this microfluidic system. And interestingly, because the distance of the chamber from the channel changes the lifetime of the RNA and the proteins, you can also see that the behavior of the circuits change. So again, remember that the longer, the faster the dilution rate. So if delta is larger, you will get a smaller concentration, a steady state concentration, and the 50 micrometer is the shortest channel, which corresponds to the shortest lifetime, and that's down here. So if you make a longer channel, then your steady state concentration goes up like you expected, but it also actually influences the time scale of these oscillations. So the oscillations become, they are fast for the short channels and they become slower for longer channels. So that's interesting. But it's also technically maybe not extremely easy for everybody because it requires some silicon micro machining and other things. So both of these fluidic approaches are technically a little more demanding. There's a question, yes. Yes, it is fixed. No, sorry, the DNA does not diffuse. The DNA stays there, so that's what they call but the RNA and the protein, they can diffuse, but they are produced all the time. So only the DNA is not produced, so that is fixed, and the others are produced. Yeah.
For a normal cell, it's a degradation because of degrading other enzymes, degrading others, or just because of thermal fluctuation.
In E. coli, the proteins are degraded, are not are mainly degraded by, no, sorry, they are sometimes degraded, but the dilution by the cell growth is the dominating thing. But RNA is degraded quickly. But here, instead of the cell growth, you just have this flushing out. So the final thing, and that's maybe the big, of course, one of the big challenges for the field and also for synthetic cells, for example, and other topics, is that these systems are not self-regenerating. So you have a lot of proteins that are required for the tRNA kind of production, or the tRNA synthetases. You need RNA polymerase, but you also need the ribosomes. And there are different ideas how to do this. And in principle, you would need to have a system that produces all of its own components, obviously. And people are not really there yet. So one thing is that, for example, you want to replicate your own DNA. So the other systems, the TXTL and the Pure, have not been used to actually also copy the DNA that you put in. And here's a paper by Hannes Mutschler and co-workers who actually put a gene for the Phi29 DNA polymerase into their cell-free reaction. And they called it transcription translation coupled DNA replication. And what you see here is this is they claim that when they add this to the Pure Express, they see some replication of the enhanced replication of the DNA by this Phi29 DNA polymerase. And what they then actually did is they put all the components that you actually, or maybe the components that you need for the production of this elongation factor, for example, and ribosomal proteins, RNA polymerase. They put all these on different plasmids, and then they looked whether these plasmids are actually also replicated in the system. And that seemed to work, but that they are not yet there. So I think I don't have time to go through all of these things here. But the point is that the idea is that they wanted to, when these systems would grow, you need to replicate the DNA that codes for all the components that need to be also produced to make more cell extract, in a sense. And this is only the replication part of the DNA. So they showed that they could put all these components on different plasmids, and that the Phi29 DNA polymerase would make copies of the DNA. And then the DNA, of course, would also be used by the Pure system to make all these components. A different approach, again, by Sebastian Merkel with Barbara Lavitzkova here, is, again, the system. You already know that. But now what they did here is they actually removed from the Pure system, they removed components. For example, like one amino acid amino acid tRNA synthetase. And then they added a plasmid to their system that could produce this missing amino acid tRNA synthetase. and they looked whether the system would still run. And the experiment looks like this here. For example, here they removed this synthetase for asparagine or for leucine. And you need to compare the green curves. They are the normal pure system, and these dark blue curves are the system where you actually added the plasmid from which the system produced this missing part. So that worked. So at least it could reproduce one part of the extract that you need. Then, of course, they did experiments where they did more. And that's maybe not a big surprise. The more you add, that actually reduces the yield. So then all the plasmids compete for the resources of the cell extract, and you need and it reduces the yield of each protein, obviously. So that's kind of a big challenge, and people need to sort of increase the activity of the extracts to be able to actually even make all these proteins. So it's one thing to just make GFP, but for the replication, you need enough kind of capacity to make 30 or 40 big components, and that's a big problem. And the other big problem, of course, is the ribosome, the holy grail maybe in the system. You need to assemble new ribosomes in the system and not just add them from the outside. And the ribosome is a complicated macromolecule containing many protein and RNA components. So you need to make all of them in your cell extract, and they need to assemble correctly in the cell extract. And apparently it's also important to figure out in which order they come together. So people study, for example, spatial organization with these DNA brushes, or maybe other ideas. So you can actually make functional ribosomes in solution. So here's a paper on bioRxiv for 1.5 years now, which has not been published, by this Japanese group, and they claim that they reconstituted ribosome self-replication. And maybe this is correct. So there is progress in the field, but it's still like a real challenge to make a system that sort of makes all of its own components for continued time. And with that, I'm at the end of the lecture. Thanks for your attention.
Sorry, can I ask?
Yeah.
Hello? Yeah.
Hello.
Can you hear me?
Yes.
Yes. Okay. I have 2 questions. The first one is related with the first part of the presentation. You mentioned that some of the goals are cell-free has some advantages that could be like less interference with different things. But I was thinking that maybe it's kind of philosophical, but we tend to use synthetic biology or biology because some processes are more efficient. For example, metabolic engineering, we can synthesize more things cheaper than in organic chemistry, right? And other kind of things. But as the time go on, some things are more efficient in different fields. We can like maybe in the future neglect or be as efficient as is in biology. But at the end, what will be the advantage of synthetic biology? Like I was thinking maybe the big advantage in biology will be like this thing that some things are autonomous, that we can make things and it can be auto-replicate. So kind of my question is, cell-free synthetic biology, it avoids the thing of interference with the different process that in the cell is happening, but we kind of lose what I think is a key component of key advantage of synthetic biology, that is this autonomous or this growing replication. So what do you think that is the key thing in synthetic biology that other things cannot have?
So you could also, well, say that maybe some people in the field, okay, because this is a lecture about synthetic biology, so I call it cell-free synthetic biology. Other people just call it cell-free protein synthesis or cell-free protein expression. So in that sense, from that perspective, it's really just a tool to make these compounds. And some biological compounds are just very difficult or impossible to make with chemistry or with standard chemistry. So I think that's maybe really more a practical consideration on, and it has not so much to do with the goals of synthetic biology or the advantages. I agree here. So I think that the point is that the cell-free systems are used for these applications, but on the other hand, they are currently still the basis for what people try to achieve in bottom-up biology, I would say, so in a sense at least. So that therefore it's kind of maybe these are tools that are shared by different communities. So the people who want to make artificial cells, they also use these systems, but then maybe to understand biology better, to make devices to create kind of some of the things that you mentioned, kind of adaptive autonomous systems and so on. That is true, but that would be maybe more also the topic of the next lectures. But the basic sort of technology that you somehow need is this cell-free expression. And of course, you can also argue whether this is there is no other solution to the problem. In a sense, I think that, of course, you would also be maybe this does not really address your question now, but I think the point is that when you say, I want to create synthetic living systems, you can also think about what would that actually require. And if you have absolutely no clue how to do it, you would just try to reconstruct biochemistry exactly how it is. And this is essentially what is done here. But okay,
Yeah.
sorry, now you had a very long comment, so I don't know.
Sorry, yeah. And also, like, I got at the end, I got the impression that the disadvantage of kind of the, if I understood correctly, the disadvantage of cell-free is that all the machinery tend to wear off. So you have to regenerate by itself. And some of the strategies kind of remind me that they are trying to reconstruct the cell, some of the components of the cell, the natural components that it's happening in the cell all the time. So maybe that this last approach is going towards like making a cell, the minimum cell with no interference. So I kind of got that impression that to avoid the disadvantage of cell-free, you have to kind of go again to reconstruct a cell to be able to be autonomous or more autonomous.
Yeah. I think that, of course, there are kind of brave colleagues in chemistry that try to do it the chemical way. So of course, you can make like high-level abstract schemes of what would be required for a system to self-replicate, and but then you need to come up with a chemistry that does these things to make that make copies of themselves, and that could potentially look different from how biochemistry looks today. But the point is that we do not have these systems, or they are less powerful, at least, as these the bacteria-based cell-free systems, for example. So I think it's always in the end some type of compromise, or it depends on what type of challenge you want to address. And so I guess that, yeah, I cannot really, for example, for me with somebody with a physics background, it's easier to do cell-free experiments than to do experiments with completely de novo synthesized chemical components, because I cannot synthesize them, but I can destroy bacteria. So that's kind of depends. So then you still hope that you can find some interesting kind of question in this, but on a fundamental level, I think you're right. It's just that nobody really knows how to do it. Nobody knows exactly how to make a self-replicating system in the lab at the moment.
Okay, yeah, that's interesting. It seems that a lot of fields in synthetic biology are towards in different approaches, but towards this end of replicating the cell or making synthetic cells, something like that.
Yeah, of course. Another question is the origin of life, which is a kind of a similar thing, but we do not have enough time to discuss.
Yeah, sorry, thank you.
But I guess that in this case, of course, you would also say, well, I just need to kickstart a basic system that is capable of replication and evolution, and then evolution does the rest for you. So I guess that really one of the main thing is that you need to have this fundamental kind of set of reactions that make a self-replicator. And as soon as you have that, then with competition for resources and some variation, Darwinian selection could kick, and then the system will find solutions that you could never come up with rationally. Hope maybe, I don't know. But this is something for the future.
Yeah. Okay. Very interesting. Thank you.
Okay. Goodbye. See you next week.